practicals. Okay, so we have just discussed about what is the definition of bioinformatics, what about the different applications of bioinformatics, what are the different tools of bioinformatics. Okay, so we have dealt with all those topics, and now we will just talk in detail about the nucleotide databases. So we will just go through a few nucleotide databases and what sort of information you can just collect through these databases. Okay. So just wait a minute. I'll just share my PPT. Okay. Okay. So this is what we will talk today about nucleotide and specialized databases. Okay, so we have discussed about that what is a database. It is actually an organized collection of related data, and the need of these biological databases is because we are dealing with all sorts of data. We are dealing with this experimental and theoretical data, and we need to have a space, a place where we can just submit and store this information, and whenever required we can just assess it, we can manage it, and we can also update it or retrieve it for different kinds of analysis processes. Okay, so where if we will just deal with sequences, then we will have these sequence databases. If we will deal with structures like protein structures, then we will have these protein data bank and other databases. So we can just go through these databases in a practical sessions, and we will also go through the formats of text file, FASTA file, and PDB. So these are the different file formats in which our information or the nucleotide and sequence protein sequences are available. So this chart we have seen in our last session also, the categories, sequences, structures, genomes, and specialized. Okay, in this session we will deal with nucleic acids, the DNA databases, GENBANK, DDBJ, and EMBL, and the specialized type of databases. Okay, the KEG, bind, and array express, and so on. Multiple of them are there in the session, and you need to understand that when the databases were created, so the reason why these different categories were being made, because we were having different sorts of information. Okay, so we were dealing with the different biomolecules, as I have talked about the different biomolecules. We were having carbohydrates, we were having proteins, we were having nucleotides. So if we had a single database dealing with all sort of biomolecules which are being submitted in a single database, then extracting and studying and managing this information would have been very much difficult. Okay, so this was the reason what made them to just go with the creation of these different types of databases. Okay, so the first one is, you can just see here, there are based on the types of data. So there is this nucleotide and protein databases. Nucleotide I have talked, like GENBANK हो गया हमारा, DDBJ होगा और EMBL होगा. All are just getting this nucleotide sequences. इन्हें आप submit कर सकते हो आराम से, और novel होना चाहिए, but you just can't go with any sort of nucleotide sequence जो already है उस database में. It should be a novel sequence. ठीक है. Similarly is the case for proteins. तो इस वजह से based on the type of data, the databases are classified into different categories. If there is source of data, so based on the source of data also, you can just go with the categorization of data. Okay, so we deal with primary data, we deal with derived databases. Okay, तो प्राइमरी डेटाबेसेस क्या होते हैं, जैसे आपके पास एक ओरिजिनल न्यूक्लियोटाइड है या आपके पास ओरिजिनल प्रोटीन है, ठीक है, और डिराइव डेटाबेसेस के एग्जांपल्स हो गए, लाइक फॉर एग्जांपल मैंने TREMBL की बात करी थी. दैट इज अ काइंड ऑफ derived databases where the proteins are being derived from nucleotide sequences, or we are dealing with these secondary type of protein databases. ठीक है, या हमारे स्पेशलाइज्ड टाइप के भी हो गए, दे आर अ सॉर्ट ऑफ डिराइव डेटाबेसेस ओनली, क्योंकि इंफॉर्मेशन जो प्राइमरी फॉर्मेट में थी हम उन्हें डिराइव करके अपना ये डेटाबेस बना रहे हैं. ठीक है, क्यूरेटेड और ऑटोमेटेड की वजह से भी आपके जो इन डेटाबेसेस हैं दे आर डिवाइडेड इनटू डिफरेंट कैटेगरीज। जनरलाइज और स्पेशलाइज तो हम पढ़ ही रहे हैं। स्पेशलाइज में हम स्पेसिफिकली न्यूक्लियोटाइड या प्रोटीन की बात नहीं करेंगे। स्पेशलाइज के केस में आपके पास बहुत एक वास्ट रेंज आ जाएगी, वेयर यू विल जस्ट टॉक अबाउट द स्पेशलाइज काइंड्स। और स्पेशलाइज काइंड्स में अभी वी हैव जस्ट डिस्कस मेटाबॉलिक भी होंगे, जीन के फंक्शन के बेसिस पर भी होंगे, वी विल हैव दिस फाइलोजेनेटिक ट्री एज वेल, जीनोम फंक्शन के बेसिस पर भी होंगे। तो ऑल सॉर्ट ऑफ these databases are there under these categories of generalized and specialized types. ठीक है। Then I got a few questions from the students that what about these databases? Can we access it easily? Publicly क्या हम access कर सकते हैं? तो majorly databases जिसकी हम बात करेंगे, we'll talk about those databases which we can just access publicly. यानी कि आप कोई भी इसे यूज कर सकता है। वी डोंट नीड टू जस्ट मेंशन एनी ईमेल आईडी, या मुझे किसी एकेडमिक इंस्टीट्यूट की इंफॉर्मेशन नहीं देनी है, या आई डोंट नीड टू पे एनीथिंग to just go and access these databases. Okay, so there are different categories, public भी है, academic भी है, commercial भी है। यानी कि अगर we are going with a certain sort of data जो कि medical data से related है और gene से related है, micro array से, तो possibly if you are dealing with such kind of databases, then it can be a paid sort of data. So you need to have a commercial ID for that. आपके पास authority होनी चाहिए that you can just go, you need to pay something to just get that data. ठीक है। एंड academic organizations जो होता है, you just need to mention कि you are a student or you are a researcher, you can just enroll yourself and you can just get your access for that particular databases. Okay, so this is what about the different categories of databases. और जो सोर्स ऑफ डेटाबेस की अभी जो मैं बात कर रही थी कि भाई अगर मैं सोर्स ऑफ डेटाबेस की बात करूंगी, तो प्राइमरी डेटाबेस हो गया मेरा प्राइमरी सोर्स। यानी कि मेरे पास एक आई एम हैविंग अमाइनो एसिड्स चेन और आई एम हैविंग अ न्यूक्लियोटाइड चेन। ठीक है, दिस इज माय प्राइमरी डेटाबेस. So what researchers are dealing with, that is said to be the primary database. It is a kind of a raw data. ये रॉ डेटा है, इसको हमने रिफाइन नहीं करा है, वी हैव नॉट मॉडिफाइड इट। ये जैसा हमने एक्सट्रैक्ट करा है वैसे वी आर ट्राइंग टू सबमिट इट इन द डिफरेंट डेटाबेसेस। एंड इट कैन बी अ डीएनए, इट कैन बी आरएनए। ठीक है, ESTs होते हैं, एक्सप्रेशन सीक्वेंस टैग्स, ये शॉर्ट सेगमेंट्स होते हैं आपके डीएनए के। एंड इससे रिलेटेड भी डेटाबेसेस है हमारे पास। प्राइमरी सीक्वेंस डेटाबेस के बारे में आई हैव टोल्ड यू, देयर इज स्विस प्रोट, जो कि particularly presently it is said to be the Uniprot knowledge base, Uniprot KB. And we have this very much famous PDB protein data bank maintained by RCSB, where you will deal with the three-dimensional structures of proteins. Okay, so all these are primary databases. The structures which are there in a PDB are being extracted they are being constructed by the help of X-ray or NMR technologies. Okay, so they are all primary only. Derived types or the secondary types which I told you, TREMTL हो गया, भी न्यूक्लियोटाइड के ट्रांसलेशन से बना है. Okay, and computational analysis, whatever kind of analysis which we are doing for our primary data, and the product which we are getting comes under the derived types. Okay, so just say for example we deal with the TREMBL, or we are dealing with the secondary type of databases where we are just going to align our protein sequences, and based on the presence of our motif regions we are getting these protein family databases like ProCite हो गया, या PFAM हो गया, print हो गया. Okay, so all sorts are under the derived types. Okay, so our topic for this session is basically nucleotide databases, and what about the specialized databases. Okay, so nucleotide databases, I just briefed you. It is a kind of database where you will have your nucleotide sequences. It is the genetic sequence database. Okay, shortly it is said to be GENBANK, and it is the one of the fastest growing repository for genetic sequences. So people, researchers who are dealing with these nucleotide sequences, what they are doing, they are just submitting their information in these databases. So whenever a researcher is going with some kind of research, so research where we are dealing with a novel sequence, we have got a new sequence, okay, which is not there in the database. So the researcher would like to just submit this sequence, okay, get an accession number, put it in a research article, and make it available to the world for the use. Okay, so this is the whole process if you are dealing with any new sequence, whether it is a nucleotide, a protein, or any other sort. Okay, so you just need to go with this sequence and you will have the page वहाँ पर जो आपका मेन वेबसाइट होता है फॉर ऑल दीज डेटाबेसेस, चाहे वो जेन बैंक हो गया, एनसीबीआई हो गया, या स्विस प्रोट या पीडीबी हो गया, you will have that particular place where you can go with the summation of your sequence. Okay, when we'll talk about GenBank database, then GenBank you will have, you will see that the files are there and the files are represented in a flat file structure. Okay, what are these flat files? You will just get to know in the future slides. But these flat files, they are easily readable by humans. We can read it, and the computers, because we need to analyze the data. So we will deal with these flat files in the form of text file or in the form of FASTA file. So the files are created in such a manner that the computer or any sort of tool or these softwares can easily read them. So this is the structure of the GenBank files. Okay, and addition to this, every file, every submission is having its particular accession number, the name is there, the classification, the whole taxonomy is there, and references. So the sequence, the whosoever had worked on that particular sequence, he must have published his or her work. So that reference should be there in the GenBank. So everything you will get there. There are certain divisions which you can see here, and then you can just see how the records are there in the GenBank. I'll show you. So this is the page for GenBank. You can see the URL here. In the URL, you can just see this is actually a part of NCBI. I told you that GenBank is a database dealing with nucleotide sequences, and what it is doing is that this is actually being maintained by the National Center for Biotechnology Information. Okay, so this is what you'll get at the home page or of your GenBank sequence database. Okay, here you can also see that here it is mentioned that it is a part of the International Nucleotide Sequence Database Collaboration, which comprises of DDBJ and EMBL and GenBank. So there is a consortium. This consortium is dealing with nucleotide sequences, the nucleotide sequence databases, and there are majorly the three databases. GenBank is there, DDBJ is there, and EMBL is there. So all these three databases, what they are doing, they are just exchanging the information within themselves. And they are dealing, all of them are dealing with the nucleotide sequences. So this will be the home page. Here you can see beside this nucleotide, we are having a taskbar here and a tab here, search. Okay, so what you need to do is if you are searching for a nucleotide sequence, like for example for humans, or you're searching for rat or mouse or any such animal or plant as well. Okay, so you can just mention the name here. This is when we are not aware about the accession number. Okay, if I want to search for a sequence, मुझे उसका accession number नहीं पता है, तो and I am generally searching for any sequence कि I want to just try to understand कि how the Homo sapiens FASTA format looks like, and what about the file for Homo sapiens sequence that is there in the GenBank database. So what I can do is मैं यहाँ पर I'll just put the name Homo sapiens here and I'll just click the button search here. So the moment I'll click the button search here, so I'll be redirected and I will get these hyperlinks. ठीक है, ये सारे जो links आ रहे हैं, they are basically representing the sequences. You can just see these are the sequences, and these sequences you can enter into any of these sequences. यहाँ पर इसका accession number दिया हुआ है. This is the accession number, and here you can see there are majorly the two tabs, the GenBank tab and the other one is the FASTA tab. Okay, so what about the GenBank tab? GenBank टैब जो है वो दिस विल गिव दैट वो फ्लैट फाइल स्ट्रक्चर जो टेक्स्ट फाइल फॉर्मेट है वो रिप्रेजेंट करेगा विद रिस्पेक्ट टू दिस सीक्वेंस, and the sequence itself that is in the FASTA format that you will deal through the FASTA tab. Okay, so this is what, Jesse, you'll click the GenBank, you'll be redirected to this particular page. You can see here is locus and the brief information. Okay, so locus ke in that same line you can just see locus. ये जो ID है AC073210, this is representing the accession number or the identification number through which you can identify this particular sequence. Okay, and the next to it is representing how many base pairs are there for this nucleotide. Then DNA is there, and what about the style of this database? It is a linear type of database. It belongs to the primates, and it was submitted back in on 9th of January 2002. So this is the basic information you can see at the locus line. Then if we are dealing with the definition, a brief definition is there, accession number again, and next is there the version. You can see that in the version there is written .8. So the version is actually representing that possibly this sequence was submitted number of times and it had those multiple number of submissions. So that is the reason this particular sequence which we are just dealing on this page is actually the 8th version for this sequence. Okay, and this belongs to the source, that is Homo sapiens. What about the taxonomy for Homo sapiens? You can just go through here. Okay, references may you'll get to know okay, what about the title of the research article in which this information is there, what about the name of the journal, and then the writer, the author of all this. So all this is there, and you can see multiple research articles are there. And then later on there is a page, I'll talk about that page. Okay, I'll talk about that page, and that I'll talk when we'll talk about the database. Okay, so here we are having this Homo sapiens FASTA format. तो आप इस FASTA format में देख पा रहे हो, FASTA format में we are having the first line, और इस first line को हम कहते हैं आपकी header line. Okay, so this is the header line. It starts with the greater than sign. There is this accession number, okay, with the version 8, name of the organism, the brief information with respect to this sequence, and the sequence starts from the second line onwards. Okay, so GATT, all this is representing the nucleotide sequence. Because this is a nucleotide sequence, you will get the codes in the form of ATGC only. ठीक है, तो सारे के सारे जो codes रहेंगे, they will be in the form of ATGC. Okay. Next database is European Molecular Biology Laboratory. It is maintained by the European Bioinformatics Institute, and this is also collecting the similar kind of information. Okay, just it is being maintained by the EBI, and the other one, the previous one, was maintained by NCBI, and both are having information, and this information is where we will just go with the DNA as well as our DNA as well as our RNA sequences, and the sequences are there submitted in the form of submissions which are related to the scientific literature as well, like the research articles or the publications. And as soon as the information is being submitted to any of these databases, whether it is GenBank, it is EMBL, or it is RDDBJ, so what will happen as soon as the submission is there, then the submission is actually being exchanged with the other databases in the consortium, the 3 of them. Okay. So the next is RTDBJ, DNA Data Bank of Japan. I had told you that this is again in the consortium, and it is working in collaboration with the other 2 again. And this one is the one that is being maintained by the Japan, and this is the full form of DNA. This DDBJ is DNA Data Bank of Japan. Okay, and this is actually back in 1986. This database came into existence, and National Institute of Genetics, this is actually maintaining this. Okay, and then This database is again dealing with all sorts of those nucleotide sequences, and these nucleotide sequences are exchanged with the other two, the GENBANK and the EMBA. Okay, so dealing with the practical session, I'll move ahead after completing my PPT. Okay, I'll show you what about these databases and how you can just go through the text file format, as well as what about this faster file format. Okay, how to retrieve that, I'll show you in the practical session. Okay, the next is my specialized type of databases. There is this metabolic pathway database, okay, and there is organism specific, gene specific, RNA specific, human gene and disease specific, then vertebrate genome. There are a lot many categories of these specialized databases, because these specialized, we are dealing with those databases which are besides a nucleotide or protein. So we are not talking about the structure of our nucleotide or the whole annotated information with respect to that nucleotide sequence, and neither we are dealing with the proteins. But besides these proteins and nucleotides, we are having a lot of biological information besides nucleotide and protein, and we are dealing with those information in this specialized type of database. Okay, so I'll start. So I told you these, under these categories, I showed you in my previous section this Kyoto Encyclopedia of Gene and Genome kit. I'll show you in the practical session again. So in the case of organism specific database, this is one of the organism specific database. So this is yeast specific database. If you want to just search any genome with respect to yeast, you can just go to this yeast genome database. Okay, so you can extract the Saccharomyces genomes through this database. Here you can see there is a quick search tab. Here is a tab. You can just mention the name, whatever you want to just search for. You can mention the accession number or name of the yeast, whatever you want to mention. You can type it here and you can click the button submit. Okay, so you will be redirected. Then there is this Arabidopsis. This is another database that is dealing with Arabidopsis, and you can extract the information. TAIR is the link, and you can deal with this database through this link. Okay, RNA sequence databases, there are actually RNAs of different types. So these many types of databases are there, and besides RNA sequence databases, we are dealing with microRNAs, then there are small RNAs, and different versions of RNAs are there. The long RNAs and the small RNAs, both are divided into various categories, and based on these categories, the researchers, they had created different types of databases. Like I'll give you an example of microRNA only. So microRNA is a kind of database. Is microRNA is something which we are having database of all sorts, with the with its function. What about its role on humans? What about its role with respect to different diseases? So we are having around thousand of databases with respect to microRNA only. So with respect to different versions, we will have different databases. And this is again a list of human mutation databases. A big list is here again. If you want to discuss or deal with any database, you want to understand what about the mutational changes that goes in a human, and what about the genes, how you can just deal with these genes, how you can extract useful information, then you can go through any of these databases. And again, I'll just say there is a very big list again, and you can find it easily on internet. Then express sequence tags, DBEST is a famous one, and this is actually a division of GenBank only. So GenBank was dealing with nucleotide sequences, but there we were talking about the whole nucleotide sequences. But when we are dealing with small segments, the expressed sequences, the tags when we are dealing with, so we can just extract them, or we can submit them in the DBEST database. Okay, and number of organisms are there in this particular database. You will have various organisms. Okay, and these organisms, the ESTs, they are submitted in GenBank, and through the entries is the system. Entries is actually the system through which you can just go and deal with the submission or the extraction of our ESTs. Okay, So I'll show you how this EST database works, okay? And what about the role of this BLAST tool, how you can deal with BLAST tool, what are the various versions of this BLAST tool as well, and what about the faster format, and so on. So we'll discuss in a practical session again. This is the transcription factor binding cited database. Here you can see, if you search it on the internet, you'll get this page. Okay, you need to just mention the DNA sequence, paste it in the FASTA format, and you will get the transcription factor binding sites. Okay, so here again we are having these single nucleotide polymorphism databases. Okay, so you all must be aware about what are single nucleotide, what is a single nucleotide polymorphism, and how we need to deal with this SNPs. Okay, so the most common form of variation in case of DNA sequences is actually SNPs. So when we will talk about different disorders, diseases, and the mutational changes, then we need to understand that we need to deal with these single nucleotide polymorphisms, and for these we can just be introduced to our database dealing with SNPs. Okay, again this is actually linked with the PubMed, the biomedical or bibliographic database, and entries will help us to retrieve the information because it is a kind of an information retrieval system. Okay, and in SNPs, you will just go through how the expression of genes are there, how they are susceptible to the diseases. Okay, what about the response of the drug on these SNPs? So, based on this information, they have created a database dealing with SNPs, and directly by submitting the SNPs, you can directly submit them. Okay, if they are approved by the organization, the authority of this database, then it will be there on the database. You can use the other information easily extracted. It is publicly available. You don't need to go with any kind of commercial registration to use this database. Okay, so another one under the category of specialized is this online Mendelian inheritance in man. And this is a kind of a comprehensive database. It is dealing with human genes and different kinds of genetic phenotypes. Okay, the full text is actually undergoing the information, information with respect to the Mendelian disorders and the different types of genes. Okay, so it's not only 12,000 genes, there are a lot many genes, and they are there in this OMIM database. And particularly this will deal with the Mendelian disorders, dealing with Homo sapiens obviously. So what about the relationship of phenotypes and genotypes, or all about the different links to different genetic sources, the different databases again. Okay, and this is developed, it was developed long back in 1995 and developed by the National Center for Biotechnology Information, NCBI, and it is actually authorized and edited at the Institute of Genetic Medicine. Okay, and Johns Hopkins University School of Medicine is taking care of this particular database, dealing with all the Mendelian disorders related to Homo sapiens. Then Unigen is one more database, and it is a set of, it is the entries, they are actually set of transcript sequences that appear to come from the same transcription locus. So okay, it can be a gene or an expressed pseudogene, and together how they are dealing with the information related to the proteins and that is the expression of genes, the cDNA clone regions, genomic location, and so on. So what about the procedure of Unigene build? What about the clustering process? All this is there under the Unigene database. So you will have the information with respect to the transcript sequences under this particular specialized type of database. Okay, so I've talked about just a few of them, the specialized type of databases. Okay, the three categories you can see here, there is interest, there is this SRS, and there is this DBGet. Okay, so interest is there. What interest is doing is, or SRS or DBGet is doing, all these three are actually helping us to retrieve the information, because these databases are not only just prepared just to submit the information should be used by the researchers all over the world. So interest and SRS, they DBGate, they are working as a medium, and through this medium we can just extract our information. Okay, so interest you can see here, this is related to, it is providing access to these number of databases. Okay, so this is helping us to retrieve the molecular biology data and the bibliographic citations as well. So bibliographic, so what is bibliographic? Bibliographic is actually when we are dealing with the research. Okay, so research as in the publications which are there, which are being published in the various journals, the articles, the review and research articles. Okay, so Entrance is linked to all these sorts of databases like PubMed is there, which I am talking about, the biomedical literature database along with Medline. It helps us to extract information from GENBANK. So it is a kind of nucleotide sequence database. Then there are protein sequence database. It helps us to extract information from protein sequences databases like UniProt is there, and TREMBL is there, PIR is there. In case of structural databases, Interest helps us to get information with respect to PDB, the 3D structure database. Then there is this complete genome assemblies when we are dealing with Ensemble or we are dealing with Tiger database. Okay, population study data sets like PopSet is there that deals with the study of population. There are data sets dealing with different populations around the world. And OMIM is there. I mentioned just back in my slides that the study of the data sets that are dealing with the Mendelian disorders, okay, related to Homo sapiens, all that information is there in this OMIM database. Okay, taxonomy, that is what about the taxonomy of different organisms in the GenBank. What about different online books, the probe sets, about the gene expression, about the microarray data sets, the 3D domains, then markers, single nucleotide polymorphism, and the conserved domains. So it is dealing with, it is helping us to just search and retrieve lot of information. Okay, and information that is divided into different categories. It can be nucleotide, it can be protein, it can be any other macromolecule, or it can be a whole genome, it can be a bibliographic or biomedical literature information. Okay, so interest is helping us for all sort of biological information, dealing with all sort of biological information. This is a page of interest, and when you search for this interest search engine, you can just see what about the PubMed, nucleotide, protein, genome structures. Not many tabs are there. And the submissions here, this is not a recent submission information. I'll show you in my practical session. So the submissions, they are actually being added every day. Okay, so people, the researchers, they are working throughout the world, and they are just submitting their research, their submissions in all these databases, adding up the information in the biological database around the world. Okay, so this is something, this PubMed is dealing with. So we have this NCBI. When you go through the URL of NCBI, you'll see there are two terms are there. There is this NIH and the other one is NLM. So NLM is actually the National Library of Medicine. Okay, so National Library of Medicine, NLM, it is actually dealing with the bibliographic databases or the biomedical literature. Okay, so biomedical literature or these bibliographic databases, the categories includes PubMed and Medline. So if you want to search any topic that has been published, the research had been done, you can just enter this PubMed. Or you want to just go with any sort of books related to your research, related to any biological system, so you can search it through PubMed, and Address helps us to search for this database. It actually links up to all these information related to articles and books. Medline is also there, that is working along with PubMed. Entrance, besides Entrance, we are having sequence retrieval system, and we also have this TBKit. So here you can see on the page of sequence retrieval system, it is a kind of a retrieval system that is searching again for several linked databases and which are there at the European Bioinformatics Institute. And similarly to Interest, it is also providing us the access to deal with different databases, and it is helping us to just go through the search. Okay, you can see here there is this Dropbox is there, and lot many things are here: nucleotide sequences, protein structures, protein families, literature, genome, mutation, metabolic pathways, biological resources. And you can type your information in the matching tab here. The tab is here. Here you can mention that if you're searching for nucleotide sequence and for a specific sequence, so you can mention here the name of the sequence like Homo sapiens. And as soon as you click here on the search button, so this SRS will just move you to the different sequence databases like GenBank, DDBJ, and EMBL. And if I just just select this protein sequence for my Dropbox and mention the name of organism, then further we will just move to different protein sequence databases like Unicode KB and PIR or TRENL. So this is the case when we deal with this information retrieval system, that is our SRS. This is the interface when you'll just enter into the page. Here you can see literature, bibliographic reference, databases. When you want to just extract, you want to go with a quick search, you can just mention the name here, and you can just select what information through which database you want to just extract the information. Okay, so for example, I want to just search for Homo sapiens. I want the literature information. I want to understand, is there any kind of Mendelian disorder related to my particular sequence, what about taxonomy. Okay, the patent abstracts also I can click here. Okay, EMBL and all sort. So I need to just click them. The moment I click them, then I'll be redirected to that particular kind of information. So this is again more information, Uniprot, other protein sequence databases, and so on. Okay, so these databases, these information retrieval systems, they are helping you. If you want, see, learning or memorizing all these databases is a very big task. Okay, so there is not only a single category, we are having a lot of information. So it will be very difficult for each and every database to be memorized. So if you are not aware about how many number of protein sequences are there, sequence databases are there, or what about the number of nucleotide sequence databases. So if we are aware, then we can go for Google search. We can just mention the name of our protein, or we can just mention the name of our nucleotide sequence database, and we can click the button search on our Google tab, and we can be redirected to that particular URL. But if we are not aware, then what we can do, we can just deal with these information retrieval systems. They will just help us to understand that how many types of databases are there, and from there we can collect our information. So these are actually helping us in this particular manner, because we are not aware about which country is maintaining which kind of database, and what database will help us to deal with what sort of information, a Mendelian disorder or a gene expression or so on. Okay, so the third database is our DBGET, like the other two information retrieval systems, and it is again integrated database retrieval system, and the major biological databases, it is categorized into KEGG databases there. Then the other databases dealing with DBGET and the searchable databases on the web, then we can just link not only the these databases, we can link other databases as well to this DBGET as soon as we go with the search. And then there are PubMed databases also. Okay, so DBGET is again a new forum for data retrieval, or the information retrieval system, and dealing with the same function, how the two of them are working. Okay, so this is the end of my slide, and I talked about basically two things. My main motive was to make you understand what are nucleotide databases. I wanted you to know that there are different specialized databases as well, and there are these three retrieval systems, the major ones, and they are actually helping you to just go with the dealing of our information retrieval, and how you can just extract the information through all this.